Good morning, everyone. Good morning, everyone. Today we are going to learn about mitosis. I'm Neha Patricia, high school biology teacher, ISG Damao. Mitosis, as we all know, is a cell division process where nucleus divides and then cytoplasm divides, leading to cell division. This process is required for growth, development, and repair in all the cells of the body. By saying so, we mean both somatic cells as well as germ cells. So, mitosis occurs in all the cells of the body and it happens throughout the lifetime of an organism for growth, development, and repair. But in unicellular organisms, mitosis is used for reproduction. This kind of reproduction is known as asexual reproduction. Mitosis, which is division of nucleus, ideally consists of 4 phases. The first phase in division of nucleus is prophase, followed by metaphase, anaphase, and finally telophase. Once the nucleus has divided, then occurs division of cytoplasm. The process is called as cytokinesis. The result of this mitotic division is formation of 2 genetically identical daughter cells. These daughter cells produced are genetically identical to each other as well as to the parent cell. Mitosis has got 4 phases which are very commonly called as PMAT phases. So prophase, metaphase, anaphase, and telophase, will be taking them one by one. Let's start with prophase. So in prophase, the chromosomes, they condense, nuclear envelope breaks down, nucleolus disappears, the centrioles, they begin to move apart and they start to form these filaments called as spindle fibers. So basically in prophase, the cell prepares for division of nucleus. Followed by prophase comes metaphase. Meta. The M here signifies that everything would come in the middle of the cell. So the spindle fibers, they get attached to the centromeres and all the chromosomes are aligned at the cell equator by these spindle fibers. All the chromosomes are aligned in the same plane at the cell equator, all the 46 chromosomes in the same plane. Then comes anaphase. The spindle fiber, they begin to shorten. While shortening, they pull the sister chromatids apart. So the sister chromatids, they reach the opposite poles of the cell. Telophase. In telophase, 2 new nuclei are formed. The spindle fiber, they disappear. And the chromosomes, they begin to uncoil and they form chromatin again. This results in finally division of nucleus and you have one big cell with 2 distinct nuclei within it. These 2 nuclei are genetically identical to each other. So after mitosis comes cytokinesis, which is the only stage which differs in plants and animal cell. In animal cell, the cell membrane, which is a very flexible covering, it simply pinches off and gets closed, resulting in formation of 2 daughter cells. But in plant cells, there is a rigid cell wall surrounding them. So, a cell plate is formed by Golgi apparatus, which thereby results in formation of cell wall and division of cytoplasm. This brings us to the end of the process mitosis. To have a better understanding of the same, here we have a video demonstrating the entire process. So let's have a look.
Mitosis begins as the long threads of DNA in the nucleus start to coil. Having already replicated, these threads emerge as the double strands we know as chromosomes. Meanwhile, protein fibers grow from the migrating centrioles, forming a latticework of spindles. For reasons scientists don't fully understand, the nuclear membrane suddenly disintegrates. With amazing accuracy, a spindle from each centriole attaches itself to each of the chromosomes. Assisted by the protein fibers, the chromosomes move center stage. In a microscopic tug of war, the spindles pull each chromatid toward opposite poles. Other spindles push against each other in a ratcheting action that stretches the cell. The chromosomes then unwind and the nuclear membranes reform. With mitosis complete, the nuclei and cytoplasm separate, creating two new cells in this ongoing cycle that sustains all life.
So that was mitosis overall, and this is what we learned.